Good day, everyone. We are the Team Ethers. I am Pichas Martinez, and together with my group mates Trisha Negro and Jasraya Quizana, are going to discuss carbohydrates. The first type of carbohydrate to be discussed is the monosaccharides. Monosaccharides is known as the simplest sugar because it cannot be hydrolyzed into smaller carbohydrates. Monosaccharides has the following characteristics. It is crystalline solid, colorless, sweet tasting, freely soluble in water, and insoluble in nonpolar solvents. Monosaccharides are used as a building blocks of a more complex form of sugars, such as oligosaccharides and polysaccharides. Also, it contains glycosidic bonds or linkages, wherein it is a chemical bond in the form of covalent connection that connects a carbohydrate molecule to another group, which can be another carbohydrate or not. And here is the general formula for monosaccharides. Monosaccharides can be classified according to the following characteristics. First is the number of atoms present in the molecule. 3 carbon is called as triose, 4 carbon tetrose, 5 carbon pentose, 6 carbon hexose, and so on. Another classification is whether they contain aldehyde groups or keto groups. A monosaccharide containing aldehyde group is called aldose, and it is called ketose when it contains keto group. So here are the structures for aldose and ketose. The aldotetrose and ketohexose are the examples when the naming for the number of carbon atoms and naming for functional groups is combined. Let us first have a practice exercise. Name the example using the carbon atom naming and functional group naming. The number of carbon atoms is 5 and the functional group present is aldehyde group. If need nyo pa ng time to answer the question, you can stop the video. Ang answer sa ating exercise is aldopentose. Going back to our discussion, we have the common monosaccharides, and they are the glucose, fructose, and galactose. Glucose and galactose have the same chemical formula. However, when we look at the percentage structure, may mapapansin ba tayong difference between the galactose and glucose? Yes, tama. So, ang difference between the glucose and galactose is the position of the hydroxyl group in the fourth carbon. Glucose is an aldose. Also, it is a product of photosynthesis in plants. Important component din siya ng amylopectin and cellulose. It also serves as an important metabolic intermediate of cellular respiration and a major source of energy. And it appears to circulate in blood, known as blood sugar. Ang fructose ay ketonic monosaccharide. Considered siya as the sweetest naturally occurring carbohydrate. Nabanggit nga natin kanina na ang glucose and galactose have similar chemical formula. However, ang galactose, generally, hindi siya nag-occur in free state. Constituent siya ng complex biomolecules. For example, ang glucose together with galactose, nagfo-form siya ng lactose. Another example is ang galactan. Example din ng monosaccharides ang ribose and deoxyribose. Ang ribose ay makikita natin sa RNA, ang deoxyribose naman ay makikita natin sa DNA. Kung mapapansin natin sa structure, ang difference nilang dalawa ay yung molecule bonded sa second carbon. Sa ribose, meron nakakabit na hydroxyl group and sa deoxyribose naman ay hydrogen atom lang. Ang deoxyribose ay mas stable siya sa ribose kasi ang ribose is more reactive and less stable siya in alkaline conditions. Before tayo mag-continue sa discussion, let's have a practice exercise. The question is, which among the following is not a monosaccharide? A. Glucose, B. Fructose, C. Sucrose, and D. Galactose. If need nyo pa ng time to answer the question, stop nyo lang yung video. Ang answer sa question is C, sucrose. Continuing again sa discussion, we have the D and L designations of monosaccharides. Glyceraldehyde is considered as the simplest monosaccharides. Glyceraldehyde contains chirality center. Also, it has two enantiomeric forms, which are known to have absolute configuration. Going back to our previous topics, the D and L is similar to the RNS IUPAC system. Ang D and L designations was first suggested by M.A. Rosanoff way before pa magkaroon ng absolute configurations ang any organic compounds. Wherein, ang ginamit nga na compound is yung glyceraldehyde na merong designated configurations. At dahil doon, yung compound na to, nag-serve siya as configurational standards for all monosaccharides. Commonly, ang monosaccharides ay merong cyclic structure. However, for conventional writing in books or in papers, the Fisher projection is used. Here, we have the example of hemiacetals and hemiketals that are written in Fisher projection. Hemiacetals are for aldoses and hemiketals are for ketoses. These generally result from the addition of an alcohol to an aldehyde or a ketone. For an instance, we see an isomeric forms of monosaccharides that differ only in their configuration about the hemiacetal or hemiketal carbon atom. It is called anomers. On the other hand, the carbonyl carbon atom is called anomeric carbon. Going back to the structural formula of monosaccharides, the Haworth perspective formula is used for the six-membered ring, since there are properties that cannot be determined using the Fisher projection. Monosaccharides is able to have mutarotation. Mutarotation is defined as the alpha and beta anomers of D-glucose interconvert in aqueous solution. For example is pyranose, which is a six-membered ring. If we will follow our discussion earlier, that a six-membered ring should use a Haworth perspective. However, pyranose is assumed to have two chair conformations. That is because pyranose is not a planar ring. The two conformations is possible because a molecule can be interconverted without the breakage of covalent bonds. As we ended the discussion for monosaccharides, we now have disaccharides. Disaccharides are the sugar formed when two monosaccharides are joined by glycosidic linkage. The most common example of disaccharide is the sucrose. Sucrose is known as a table sugar and it is composed of glucose and fructose. The encircled part of the structure is the glucose. And for this encircled part, it is the fructose.
Hello everyone, I am Keisha from Team Ethers and I will be discussing about polysaccharides. First, let's discuss the etymology of the word polysaccharides. The word poly came from the Greek word polus meaning many, while saccharides from the word saccharon meaning sugar. Polysaccharide is also known as glycan. We have learned from the previous discussion that a monosaccharide is comprised of a single simple sugar unit. For example, we have glucose, fructose, or galactose, and they cannot be broken down into simple sugar units. These three monosaccharides are combined in various ways to make more complex carbohydrates. Meanwhile, the disaccharides are comprised of two monosaccharides which are bonded together. Most carbohydrates found in nature occur as polysaccharides, polymers of medium to high molecular weight. Polysaccharides are different from each other in the identity of their recurring monosaccharide units. in the length of their chains, in the types of the bond linking the units, and in the degree of branching. Understanding polysaccharides is a complex matter, and in fact, we can further elaborate polysaccharide by understanding its two types, which are homopolysaccharide and heteropolysaccharide. So, when we say homopolysaccharide, these are the polysaccharides that are polymers of a single monosaccharide. Those made up of more than one type of monosaccharide are called heterosaccharides. Some homopolysaccharides serve as storage forms for monosaccharides that are used as fuels. For example, meron starch and glycogen. And other homopolysaccharides gaya ng cellulose and chitin, nagsaserve naman sila as structural elements in plant cell walls and animal exoskeletons. On the other hand, heteropolysaccharides, they provide extracellular support naman sa mga organisms for all kingdoms. For example, yung rigid layer ng bacterial cell envelope, gaya ng peptidoglycan, that is composed in a part of a heteropolysaccharide built from two alternating monosaccharide units. So, paano nga ba mas madaling madidistinguish yung difference from homopolysaccharides and heteropolysaccharides? So, pag sinabi natin homopolysaccharides, sinabi ko naman din kanina, from the word homo, means one. So, yung sugar na present dun sa structure is the same all throughout the compound. Meanwhile, for the heterosaccharides, there may be two or more presence of different sugars um, bonded together. And furthermore, each of these either of these polysaccharides, homo or hetero, um, they both have branch and unbranched types. Now let's further discuss homopolysaccharides. Homopolysaccharides are also classified on the basis of their monosaccharide units. So, for example, a homopolysaccharide consisting of glucose monomeric units is called glucan. Before we further proceed to the other examples of homopolysaccharides, let's first have a short and quick activity. So, can you name what compound or what homopolysaccharide is shown in the slide? So, I'll give you 5 seconds. Five, four, three, two, one. If you answered galactose, then you are correct. Shown in the slide is a homopolysaccharide consisting of galactose monomeric units. So the question is, what do we call to these monomeric units? If your answer is galactan, then you are correct. Now let's move forward with the discussion of homopolysaccharides that are stored forms of fuel and serve structural roles. The three main important glucan polysaccharides are starch, cellulose, and glycogen. Let's first discuss about starch. It occurs as microscopic granules in the roots, tubers, and seeds of plants. So, yung commercial sources of starch includes corn, potatoes, wheat, and rice. Kung i-heat yung starch with water, possible na mag-cause ng swelling sa granules and mag-produce ng colloidal suspension kung saan yung two major components can be isolated. So, one fraction na makukuha mo from that process is the amylose, which is about 10 to 20%. And amylose typically consists of more than 1,000 D-glucopyronoside units that is connected with alpha linkages between C1 of one unit and C4 of the next. So from that process, yung another component na possible ma-isolate is the amylopectin, which is about 80 to 90%. And yung chains ng amylopectin are branch and it consists of hundreds of interconnecting chains of 20 to 25 glucose units. Starch also have a vital functions in our body. The first one is as an energy source. Our body kasi binibreak down niya yung starch into glucose to serve as a fuel or energy source. And meron ding type of starches that are resistant, or these are basically called the resistant starches, which they pass undigested into our colon. And yung mga starches like this, they serve as dietary fiber, and they provide nutrition for the beneficial bacteria in our colon and keeping them thriving and healthy. Another is glycogen. Yung structure niya is gaya ng sa amin lepectin, pero mas maraming branch yung kanya. So, kita na, as shown in the slide, yung glucose, similar siya to glycogen. So, glycogen is a homopolysaccharide. The localization of glucose units within a large highly branched structure simplifies one of the cell's logistical problems, that of having a ready source of glucose when cellular glucose concentrations are low, and of being able to store glucose rapidly when cellular glucose concentrations are high. So there are enzymes within the cell that catalyze the reactions by which the glucose units are detached from or attached to glycogen. Animals store energy as fats in the form of triacylglycerols as well as glycogen. And glycogen is the main storage polysaccharide of animal cells. Another example of a homopolysaccharide is cellulose. It is a fibrous, tough, and water-insoluble substance that is found mostly in cell walls of plants, and particularly in stalks, stems, trunks, and all the woody portions of plant body. So when we examine the structure of cellulose, we find another example of polysaccharide in which nature has arranged monomeric glucose units in a manner that suits its function. So cellulose contains the glucopyronoside units linked in 1 to 4, fashioned in a very long unbranched chains. Unlike starch and glycogen, however, the linkages in cellulose are biglycosidic linkages. So kapag ini-ingest natin yung glycogen and starch, na-hydrolyze sila ng alpha amylases and glycosidases. Yung enzymes sa saliva and also sa intestine na nagbe-breakdown ng alpha 1 to 4 glycosidic bonds between glucose units. So, karamihan ng mga vertebrate animals, hindi nila nadadigest or natutunaw yung cellulose as a fuels or nagagamit yung cellulose as fuel source. Kasi kulang sila dun sa enzyme na nag-hydrolyze ng beta 1 to 4 linkages. So, yung termites, kaya nilang mag-digest ng cellulose and also ng wood because yung intestinal tract nila ay merong symbiotic microorganism known as trichonympha na nagsisecrete ng cellulase na nag-hydrolyze ng beta 1 to 4 linkages. Chitin is a linear homopolysaccharide composed of N-acetyl glucosamine residues in beta 1 to 4 linkage, and the only chemical difference from cellulose is the replacement of hydroxyl group at carbon 2 with an acetylated amino group. Chitin forms extended fibers similar to those of cellulose, and like cellulose cannot be digested by vertebrates. Chitin is a principal component of the hard exoskeletons of nearly a million species of arthropods, or insects, lobsters, and crabs, and is probably the second most abundant polysaccharide next to cellulose in nature. An estimated of 1 billion tons of chitin are produced each year in the biosphere. Now, let's proceed with the discussion of bacterial and algal cell walls contain structural heteropolysaccharides. Certain marine red algae, including some of the seaweeds, have cell walls that contain agar, a mixture of sulfated heteropolysaccharides made up of D-galactose and an L-galactose derivative ether linked between C3 and C6. Agar is a complex mixture of polysaccharides, all with the same backbone structure but substituted to varying degrees with sulfate and pyruvate. So agarose is the sugar component with the fewest charged groups, such as few sulfates and pyruvates. The remarkable gel-forming property of agarose makes it useful in biochemistry laboratory. When a suspension of agarose in water is heated and cooled, the agarose forms a double helix. Two molecules in parallel orientation twist together with a helix repeat of three residues, water molecules are trapped in the central cavity. These structures in turn associate with each other to form a three-dimensional matrix that traps large amounts of water. Now let us discuss glycosaminoglycans are heteropolysaccharides of the extracellular matrix. So extracellular matrix or ECM is also known as ground substance. So the ECM holds the cells together and provides porous pathway for the diffusion of nutrients and oxygen to individual cells. So let me show you a diagram. So makikita niyo sa diagram, yung ECM nasa labas siya ng plasma membrane. And it is composed of various materials that forms a matrix, which includes fibronectin, laminin, collagen fibers, elastin fibers, integrins, ion channels, and proteoglycans. Proteoglycans are macromolecules of the cell surface of extracellular matrix in which one or more sulfated glycosaminoglycan chains are joined covalently to a membrane protein or a secreted protein. So short overview na lang for glycosaminoglycans. They are the family of linear polymers composed of repeating disaccharide units. So as shown in the slide, there are two different monosaccharide for every disaccharide, and they provide viscosity, adhesiveness, and tensile strength to the extracellular matrix. So this is the summary. So we have for polysaccharides, we can divide it to two types, which are homopolysaccharides and heteropolysaccharides. So homopolysaccharides examples are starch, glycogen, cellulose, and chitin. And for heteropolysaccharides, we have agar, agarose, glycosaminoglycans.
The next topic would be about the glycoconjugates, more specifically about the proteoglycans, glycoproteins, and glycosphingolipids. Polysaccharides and oligosaccharides serve as stored fuels such as starch, glycogen, and dextrin, and structural materials such as cellulose, chitin, and peptidoglycans. But they also act as information carriers. As information centers, some provide communication between cells and their extracellular surroundings. Others label proteins for transfer to and localization in specific organelles or for destruction when the protein is malformed. And there are some that serve as recognition sites for extracellular signal molecules like growth factors, or extracellular parasites such as bacteria or viruses. Um, Oligosaccharide chains attached to the plasma membrane form the glycocalyx, which plays a crucial role in cell-to-cell recognition, adhesion, migration, blood clotting, immune response, wound healing, and other cellular processes. Informational carbohydrates are often covalently joined to proteins or lipids to form glycoconjugates, the biologically active molecules. The image shown is the glycoconjugate. It's just the structures of some typical proteoglycans, glycoproteins, and glycosphingolipids. Proteoglycans are macromolecules of the cell surface or extracellular matrix, in which one or more sulfated glycosaminoglycan chains are joined covalently to a membrane protein or a secreted protein. The glycosaminoglycan chain can bind to extracellular proteins through electrostatic interactions between the protein and the negatively charged sugar moieties on the proteoglycan, and they are also a major components of all extracellular matrices. Glycoproteins, on the other hand, have one or several oligosaccharides of varying complexity joined covalently to a protein, and they are usually found on the outer face of the plasma membrane as part of the glycocalyx, in the extracellular matrix, and in the blood. They are also found in specific organelles such as Golgi complexes, secretory granules, and lysosomes. The oligosaccharide portions of glycoproteins are very heterogeneous, and like glycoaminoglycans, they are rich in information, forming highly specific sites for recognition and high affinity binding by carbohydrate binding proteins called lectins. Glycosphingolipids are plasma membrane components in which the hydrophilic head groups are oligosaccharides. As in glycoproteins, the oligosaccharides act as specific sites for recognition by lectins. The brain and the neurons are rich in glycosphingolipids, which help in nerve conduction and myelin formation. Glycosphingolipids also play a role in signal transduction in cells. Next, we'll be delving into our proteoglycans, wherein proteoglycans are glycosaminoglycan containing macromolecules of the cell surface and the extracellular matrix. Proteoglycans act as tissue organizers and influence cellular activities like growth factor activation and adhesion. Basic unit consists of a core protein with covalently attached glycosaminoglycans linked via a tetrasaccharide bridge to a serine residue. The serine residue is typically in the sequence serine glycine X glycine or ser gly X gly, where X is any amino acid residue. Here in the figure shown is a proteoglycan structure, which showcases the tetrasaccharide bridge. Here it shows a typical tetrasaccharide linker, the one shaded in blue, which connects a glycosaminoglycan. Um, Here it is chondroitin for sulfate, which is shaded, the one shaded in orange, to a serine residue in the core protein. Here, the xylose residue at the reducing end of the linker is joined by a glycosidic bond. Many are secreted into the extracellular matrix, while some are integral membrane proteins. The sheath-like extracellular matrix, such as the basal lamina, contains core proteins with several covalently attached heparan sulfate chains. Now, there are two major families of membrane heparan sulfate proteoglycans, and these are the syndicans and the glypicans. For the syndicans have a single transmembrane domain and an extracellular domain with 3 to 5 chains of heparan sulfate and sometimes chondroitin sulfate. Here in the figure shown in the syndican part, they are held in the membrane by hydrophobic interactions between a sequence of nonpolar amino acid residues and plasma membrane lipids. They can be released by a single proteolytic cut near the membrane surface. And in the typical syndican, the extracellular amino terminal domain is covalently attached by the tetrasaccharide linkers, such as the one shown in the previous slide, to 3 heparan sulfate chains and 2 chondroitin sulfate chains. Moving to glypicans. Glypicans are attached to the membrane by a lipid anchor, a derivative of the membrane lipid phosphatidylinositol, and the glypicans are held in the membrane by a covalently attached membrane lipid, but are shed of the bond between the lipid portion of the GPI anchor, as shown in the image in the right, and the oligosaccharide linked to the protein is cleaved by a phospholipase. All glypicans have 14 conserved cysteine residues, which form disulfide bonds to stabilize the protein moiety, and either 2 or 3 glycosaminoglycan chains attached near the carboxyl terminus close to the membrane surface. Both syndicans and glypicans can be shed into the extracellular space, regulated by proteases and phospholipases. Shedding allows cells to quickly change their surface features and is activated in proliferating cells like cancer cells. Proteoglycan shedding plays roles in cell to cell recognition, adhesion, proliferation, and differentiation. Various chondroitin sulfate and dermatan sulfate proteoglycans exist, but some membranes bound and others secreted into the extracellular matrix. Glycosaminoglycan chains bind to the extracellular ligands, affecting their interaction with cell surface receptors, and heparan sulfate has structured domains that bind specific proteins and signaling molecules, altering their activities. Mechanisms of action include inducing conformational changes in proteins, enhancing protein-to-protein interactions, acting as cor- co-receptors for growth factors, and maintaining high local concentrations of molecule in the cell surface. Following are the four types of protein interactions with Venice domains of heparan sulfate. Here, the glycosaminoglycan chains can bind to a variety of extracellular ligands and thereby modulate the ligand's interaction with specific receptors of the cell surface. The NS domain stands for highly sulfated domains, and they alternate with domains having unmodified GLC-NAC and GLCA residues, or N-acetylated or Na domains. Here, the heparan sulfate molecules with precisely organized NS domains, or highly sulfated domains, bind specifically to extracellular proteins and signaling molecules to alter their activities. The change in activity may result from a conformational change in the protein that is induced by the binding, as seen here in this figure. A conformational change induced in the protein antithrombin AT uh, on binding a specific pentasaccharide NS domain allows its interaction with blood clotting factor Xa, preventing clotting. It may be due to the ability of adjacent domains of heparan sulfate to bind to two different proteins, bringing them into close proximity and enhancing protein-to-protein interactions, as seen here in the figure B, which is enhanced protein-to-protein interaction. Here, the binding of AT and thrombin to two adjacent NS domains brings the two proteins into close proximity, favoring their interaction, which inhibits blood clotting. A third general mechanism of action is the binding of extracellular signal molecules, growth factors, for example, to heparan sulfate, which increases their local concentrations and enhances their interaction with growth factor receptors in the cell surface. In this case, the heparan sulfate acts as a co-receptor. Um, it says in the image, and as domains interact with both the fibroblast growth factor or FGF and its receptor, bringing the oligomeric complex together and increasing the effectiveness of a low concentration of FGF or fibroblast growth factor. Finally, in the last type of mechanism, the NS domain interact electrostatically with a variety of soluble molecules outside the cell, maintaining high local concentrations at the cell surface as seen in the figure D, cell surface localization or concentration. It is stated in the image that the high density of negative charges in heparan sulfate attracts positively charged lipoprotein lipase molecules and holds them by electrostatic and sequence-specific interactions within its domains. And the importance of correctly synthesizing sulfated domains in heparan sulfate is demonstrated in mutant knock, quote unquote knockout mice lacking the enzyme that sulfates the C2 hydroxyl of iduronate or IDOA. Some proteoglycans can form proteoglycan aggregates, which are enormous supramolecular assemblies of many core proteins, all bound to a single molecule of hyaluronan. And these are contributing to tissue development and resilience. And aggrecan, which is associated with hyaluronan, interacts strongly with collagen in cartilage, enhancing its strength and resilience. Fibrous matrix proteins like collagen, elastin, and fibronectin form a cross-linked meshwork, providing strength and resilience to the extracellular matrix. And these interactions between cellular and extracellular molecules create a complex network influencing cell matrix interactions. And the image on the immediate right is the proteoglycan aggregate of the extracellular matrix, which is a drawing of a proteoglycan with many aggrecan molecules. And each aggrecan molecule contains many covalent bound chondroitin sulfate and keratin sulfate chains. With these link proteins at the junction between each core protein at the hyaluronan backbone mediate the core protein to hyaluronan interaction. Now the image on the far left is the interactions between cells and the extracellular matrix, and the association between cells and the proteoglycan of the extracellular matrix is mediated by a membrane protein or integrin and by an extracellular protein or fibronectin with binding sites for both integrin and the proteoglycan. Now here, glycoproteins are carbohydrate protein conjugates with smaller, more diverse glycans than proteoglycans. And the carbohydrates attached to the serine or the threonine residues, which are O-linked or asparagine residues, or the N-linked. As seen here in the figure on the left side, O-linked oligosaccharides have a glycosidic bond to the hydroxyl group of the serine or the threonine residues, which are shaded in light red. And the here is illustrated here with galnac as the sugar at the reducing end of the oligosaccharide. One simple chain and the one complex chain are shown. Now here in figure B, N-linked oligosaccharides have an N-glycosyl bond to the amide nitrogen of an asparagine residue, just here shaded green, and here illustrated with GLCNAC as the terminal sugar. Three common types of oligosaccharide chains that are N-linked in glycoproteins are shown. Glycoproteins may have one or multiple oligosaccharide chains ranging from 1% to 70% or more of the protein's mass. N-linked oligosaccharides are typically found in the consensus sequence, NPSD, but not all potential sites are used. O-linked oligosaccharides lack a specific consensus sequence but tend to occur in regions rich in glycine, valine, and proline residues. Certain glycoproteins in the cytoplasm and nucleus carry single N-acetylglucosamine residues reversibly attached to serine residues. And mucins mucins are secreted or membrane glycoproteins containing numerous O-linked oligosaccharide chains, imparting slipperiness to mucus. Glycomix involves characterizing all carbohydrate components of a cell or tissue, including those attached to proteins and lipids. It is the determination of the full complement of sugar-containing molecules in a cell or tissue, and the determination of the function of each such molecule. Many eukaryotic secreted proteins, such as blood proteins, examples are immunoglobulins, hormones, and milk and pancreatic proteins are glycoproteins. Lysosomal proteins are typically also contain glycans. Glycoproteins are not the only cellular components that bear complex oligosaccharide chains. Some lipids too have covalently bound oligosaccharides, such as the gangliosides, and are membrane lipids of eukaryotic cells in which the polar head group, the part of the lipid that forms the outer surface of the membrane, is a complex oligosaccharide containing a sialic acid, and the other monosaccharide residues. Some of the oligosaccharide moieties of gangliosides, such as those that determine human blood groups, are identical with those found in certain glycoproteins, which therefore also contribute to blood group type. Like the oligosaccharide moieties of glycoproteins, those of membrane lipids are generally found on the outer face of the plasma membrane. Now, the lipopolysaccharides, or LPS, are the dominant surface feature of the outer membrane of gram-negative bacteria such as Escherichia coli and Salmonella typhimurium. These molecules are prime targets of the antibodies produced by the vertebrate immune system in response to bacterial infection and are therefore important determinants of the serotype of bacterial strains. The lipopolysaccharides of Salmonella contain six fatty acids bound to two glucosamine residues, one of which is the point of attachment for the complex oligosaccharide, which is shown in the image to the right. E. coli has similar but unique lipopolysaccharides. The lipidate portion of the lipopolysaccharides of some bacteria is called endotoxin. Its toxicity to human and other animals is responsible for the dangerously lowered blood pressure that occurs in toxic shock syndrome, resulting from gram-negative bacterial infections. Now let us recall: what is the determination of the full complement of sugar-containing molecules in a cell or tissue, and the determination of the function of each such molecule? The answer is glycomics. Moving on to carbohydrates as informational molecules, the sugar code. Lectins are proteins binding carbohydrates specifically and with moderate to high affinity, play crucial roles in cell-to-cell recognition, signaling, adhesion, and intracellular protein targeting. Plant lectins are abundant in seeds, are likely deter insects and predators, while purified plant lectins are valuable tools for detecting and separating glycans and glycoproteins in laboratory settings. Cell surface lectins initiate cell-to-cell interactions, with oligosaccharide tags governing degradation rates of certain peptides, proteins, and blood cells in vertebrates. Lectins are also implicated in human diseases and infectious agent interactions. Selectins is a family of cell surface lectins, and they facilitate cell-to-cell recognition and adhesion, such as leukocyte movement through capillary walls during an infection or inflammation. At the infection site, P-selectin on the surface of capillary endothelial cells interacts with a specific oligosaccharide of the surface glycoproteins of circulating leukocytes. Uh, this interaction slows the leukocytes as they roll along the endothelial lining of the capillaries. A second interaction between integrin molecules in the leukocyte plasma membrane and an adhesion protein on the endothelial cell surface now stops the leukocyte and allows it to move through the capillary wall into the infected tissues to initiate the immune attack. And now two other lectins participate in this quote-unquote lymphocyte homing. These are the E-selectin on the endothelial cell and L-selectin on the leukocyte, and they bind their cognate oligosaccharide on the leukocyte and endothelial cell, respectively. Human selectins mediate the inflammatory responses in rheumatoid arthritis, asthma, psoriasis, multiple sclerosis, and the rejection of transplanted organs. And thus, there is great interest in developing drugs that inhibit selectin mediated cell adhesion. Many carcinomas express an antigen normally present only in fetal cells that, when shed into the circulation, it facilitates tumor cell survival and metastasis. Additionally, pathogens including bacteria, viruses, and parasites adhere to animal cells via lectin. Carbohydrate binding with X-ray crystallography revealing the molecular complementarity underlying lectin-carbohydrate interactions. Now, let us recall. Um, these are useful reagents for detecting and separating glycans and glycoproteins with different oligosaccharide moieties. The answer is purified plant lectins.
For the last topic, we have working with carbohydrates. Establishing the complete structure of oligosaccharides and polysaccharides requires determination of linear sequence, branching positions, and configuration of each monosaccharide unit, and the position of the glycosidic linkages, which is a more complex problem than protein and nucleic acid analysis. And the structures of oligosaccharides and polysaccharides are usually determined by a combination of methods, specific enzymatic hydrolysis to determine stereochemistry at the glycosidic bond and to produce smaller fragments for further analysis, methylation to locate glycosidic bonds, and stepwise degradation to determine sequence and configuration of anomeric carbons. So also we have mass spectroscopy and NMR, which is high resolution, which is applicable to small samples of carbohydrate only, and yield essential information about sequence configuration and at anomeric and other carbons and position of glycosidic bonds. So these are the things we should have some overview or few overviews before we work with carbohydrates. And that will be all for our presentation. Thank you for listening.